Welcome to this new tutorial for the ivma3 analysis software. In this tutorial, we'll be talking about the motile fraction. So the motile fraction is a useful concept or a useful quantifier of in vitro motility, which basically tells you not how fast the filaments are moving, but it tells you how often the filaments are moving. So for some experiments, if you apply conditions in the experiment, you can see that the effect actually would be that filaments are stopping more often than in the control condition, or they're moving more often, while the velocity while they're moving is not very strongly affected. And in situations like this, you would want to look at the motile fraction of the filaments. And this tutorial will explain you how you can get this from video files that you recorded in experiment. You will see that the interface today looks slightly different, say over here. Those of you who use the same operating system, this is on a MacBook, so we're working on OS X today. But in MATLAB, that shouldn't make too much of a difference. It should work pretty much the same on all platforms, so don't be too confused about this. Don't worry too much about this. Where we're going to start is by analyzing the example video files that you get with your ivma3 if you download it from the GitHub repository. And you can see here in example video files we have 3 slides with videos. And what we are going to do is to first analyze these slides here, and then determine the motile fraction for each of the separate slides to show you how you can calculate it from your own experimental data as well. So the first thing would be to go and start the analysis launcher here and run it. In here I explained this at length in a tutorial before. We should pick a source, example results, no example video files, this one here, which contains the video files that we want to analyze, so they're all going to be processed in a batch. And we also take a target folder I created before here, motile fraction analysis and analysis results, so all the results will be put into this folder. Other than that, I will basically keep the standard parameters here. You have to watch out with this one. I pointed it out in another tutorial before. You have to adjust the magnification factor to your own very experiment, or your velocity values are going to be way off and you get misleading results. So I think we're ready to go. I'm going to start the analysis. You can see here on the command line how things are getting into gear. And we should probably see the first results, the first reports coming up on the command line here soon. So now that the analysis is completed, I just stopped the video recording for the time being. You can see here how long the computation actually took. So a little less than 4 minutes, that's how long I was gone. Now we have analyzed the videos, so okay here. And we should probably quickly take care of the quality control. So we're going to use the trace dropper to get rid of bad traces in there and get the data ready for determining the motile fraction from them. So let's get the trace dropper running here. Choose a batch to load up. And here, and you want this trace dropper, this file. And okay, the important ones are space, the space button or space key, space bar, and backspace to get rid of them. And then we're going to quickly go through these filaments here. So if it is entirely unclear what I'm doing here, you have to go back to one of the earlier videos where I explained how the trace dropper is working. It's basically making sure that we don't have any traces where filaments have crossed or have gotten stuck on the surface and started to turn into a rotor or like a swirling filament. We have to make sure that these are not present in our data, and I'm just very quickly getting rid of them here. And I am recording it and keeping it in the video in case you need more examples of what kinds of filaments we typically keep and reject in our lab. While I have to say that all this is always a little bit of a question of your own eye measure with this. So there is no 100% solid rule how to reject these, but say if you see something like too curly or clearly crossing over where you can see two filament shapes, then you might want to get rid of this. Like here, for example, that should go. You can keep this. Like here, you see two filaments crossing. They really have to go. And for some, they're just straight like here, so it's a pretty clear case. But everything in between, you just should be consistent in how you reject them and keep them. Other than that, there's a little bit of freedom in how to do this. You just have to find a standard for yourself or for your lab that you go with. And you can use my choices here that you can see tracked on the right here as an orientation if you so wish, or you need some hints as to what to keep and what to reject. And I'm just going to go through this more or less quickly, and the less I talk about it, the quicker I can deal with this. So I'm going to be quiet for a minute or two here, while you can still watch how I'm going through this data set, getting rid of the bad traces in here. As said, you can follow my decisions in the bar on the right, so I don't have to say for each and every single trace here if I kept it, if I dropped it, but you can basically just see it as I am going along. You can also see here, there's the batch file 7 of 9, so here. And this tells you how much of your data set you have actually dealt with, so there's not very much left, so let's just quickly get through this, guys. Oh, that one was not so great. I kept it, but that one should have gone. So luckily now we have this undo button here that allows you to undo the last trace, the choice for the last trace. Sure, everyone is happy about the undo button. I am. And I think we're in the last slide to take care of here. So not far to go anymore. Okay, batch finished. Here we are. And now we're ready to actually get the motile fraction out of these guys. The actual calculation of the motile fraction we will now do with the module collect result files, which also was seen in prior tutorial videos. So for some of you it might actually be a familiar sight. So here the first thing is that we need to get result files into this. And for this we're going to locate the motile fraction analysis results, slide 1. Basically you get all the analysis results into this module to work with. One up here, so that we can first determine how exactly we're going to calculate the motile fraction, and then calculate it separately after that for each of the slides. Okay, now we should have 1, 2, 3, 4, 5, 6, 7, 8, 9 different analysis results here for 3 slides. Each of them has 3 videos in it, so 9 analysis result files here is seems to be the correct number, so we're done selecting them. And now what's new in here for determining the motile fraction is this V threshold, or threshold velocity, which we use to separate the instantaneous or frame-to-frame sliding velocities of actin into active motion above threshold, or into actin that is currently arrested below the sliding velocity threshold. So first we have to set this threshold here, and for this we should plot a VL diagram of all these slides put together. So let's calculate the values and look at this graph here, and you can see, well, here's the actin length and here's the actin velocity. You can see that there's this like really nice population here, and extending to over here to the right, which exhibits active sliding of the actin filaments. But there's also this part here which reaches more towards towards zero velocity, so towards arrest to the motility surface. So what we need to do is to find a value of velocity that will act as a threshold between the active and the arrested population here. And looking at this graph, I would say a good point to cut would be here. So let's pick 0.2, or maybe to be a little bit more accurate, a little bit higher, 0.225. So, no, not here. Here, let's put 0.225 into here, and that should actually give us a motile fraction now if we hit calculate values. Now you can actually see in here the threshold velocity that we picked is put into the graph here and into the graph here now, so that you can see where you actually apply the threshold. And so far it seems like it actually separates this part of the motility and this part of the motility quite well. So you can see this in the graphs. But also you can now go here and see what the motile fraction in your motility assay here has been. And 0.9 means 90% of the time that actin was actually observed, it has been actively moving as per this threshold that we set before. And that's a relatively high number, and looking at these graphs here also the motility seems to be working fairly well. But maybe to make your quantification a little bit more sensitive, you want to go into a range where you can already see that the motile or the filament motion sometimes is stopping. So this would be maybe in a range from 0.5 to 1.5 micrometers here. So let's put these limiters in, 0.5 to 1.5. And again, calculate the values and get the graphs out of that. And so now you can see that we have a limiter here, and within this range we determined with this threshold the motile fraction. And it's only slightly lower here. So still, even though we're looking in this region here, it seems that filament motion is quite persistent in here. So the important thing is that now we have set a length range, so we're actually controlling the length of filaments that we're actually looking at. And also we have set the threshold velocity here. And actually, in terms of the length control, sometimes there are effects of having filaments breaking up, and then you would get especially filaments in the length below 1 micrometer. And just because you have more, it might seem like you have an effect, even though all that is happening in your experiment is that they're breaking up. I'm going to make the range in which we determine the motile fraction even more narrow, so these effects are counteracted. Okay, so now we have this range here where we determine the motile fraction, and I think we're pretty well set up to start analyzing the slides separately, which is basically what we're going to do now, keeping these parameters the same across all of the slides, so those have to be consistent. So now we would go and open here, only the results for, say, the first slide. So no, that was the wrong one. Remove. Here, and here we have all the 3 videos in there. See the 3 here. And then we go ahead and actually calculate the values. You can see less filaments, of course, and now we can see the motile fraction from here, 0.91, so forth. Let's do the next slide, slide 2. Get the analysis results in here. Done, and calculate the values. Now we have 0.88. Doesn't seem largely different from what we had before, to be honest. And let's do the last slide here, slide number 3. Get the analysis results in there. No, again, the wrong one. Remove. And here, for some reason, this is very small on the MacBook screen. In any case, here's the third one. We calculate the values again, and again, quite consistent. We are very close to what we had before in terms of the motile fraction. You can see it here. And so with this, we have calculated the motile fraction for these 3 videos. We've gone from analyzing the raw video material to via the quality control to reject traces that were not okay, to finding the analysis parameters for determining the motile fraction, taking into consideration the whole data set, and then going into the different parts or the different flow cells contained within the data set to determine the motile fraction per each flow cell, which is actually the number that in the long run you, or in the end of an experiment, you would want to calculate. So I hope this tutorial has given you enough detailed description of how to calculate the motile fraction, so that you can easily go ahead and do this yourself with the software. But if there are questions, then please don't hesitate to ask. If there are suggestions how to improve it, then also please come forth with it, and we'll see if we can implement them. And we're also always happy to hear if someone has used this for a study and it has actually benefited them in their own work, because this is part of why we made this software. So with that, have fun with it, good luck, and that's it for today.